So to talk about genetics, we first need to talk about the structure of DNA. So thus far, we've talked about nucleic acids. So we've covered what a nucleotide is, the monomer of DNA. So we should remember that every nucleotide contains a sugar, a 5-carbon sugar, a phosphate group, and a nitrogenous base. At the bottom of the sugar, which is deoxyribose in DNA, we have a 3' carbon attached to a hydroxyl group. And at the 5' carbon, we have our phosphate group. Our nitrogenous base projects inward towards our complementary DNA strand. When we're thinking about our nitrogenous bases, we have 4 options: thymine, adenine, cytosine, and guanine. And they form hydrogen bonds in specific pairings. And specifically, thymine on one strand will pair to adenine on the complementary strand. Guanine on one strand pairs to cytosine on the complementary strand. The two strands, the two complementary strands, are anti-parallel, which is to say, if we're looking at one strand, we'll have a free phosphate group at one end. And on its complementary strand, we see that the nucleotides are basically upside down. So we don't have a phosphate group. Instead, we have our hydroxyl group. And the free phosphate group, the 5' end, is on the other end of the DNA molecule. So when we're packing DNA into a cell, we actually have to package it much more tightly than just one linear strand. DNA is really long, it's really big. To fit it into a cell, we need to basically tightly spool it together to cram it all into a nucleus. So DNA kind of has different levels of structure. The first are our nucleotides, right? Our monomers, which are building blocks of DNA and also RNA. If we're talking about RNA nucleotides, we create covalent bonds between our nucleotides. And that gives us a strand. So we have a linear strand of DNA. That's our polymer. The strand has a complementary strand, and together they form a double helix. So they kind of twist around each other. That's our 2 strands of DNA, which is our typical shape of a DNA molecule. And from there, the DNA winds around a series of proteins, most of which are called histone proteins, into structures called chromosomes. So chromosomes are DNA coiled tightly around a bunch of proteins. If we're referring to all our chromosomes together, we call that the genome. As we talk about DNA, we are also going to want to distinguish the sort of different states it can be in. So our cells are going to exist in different states of division, different states of function, and the DNA is going to look different in cells that are doing different stuff. So DNA usually exists as something called chromatin, which is a combination of DNA and proteins, so like our histone proteins. And the DNA is kind of compact, but it's looser than a really tightly wound chromosome that we'll see as we go into mitosis, which we'll talk about later. Okay, so chromatin is a complex of DNA and proteins that compacts the DNA to fit enough into the cell, but is loose enough to allow different enzymes to access that DNA so that they can read specific genes to make proteins, so that they can make new copies of the DNA, so that they can do stuff. On our DNA, we're going to have individual genes, which we'll talk about in a second. The other thing to know is that during mitosis, our chromatin condenses into chromosomes, which are highly condensed, so they're wrapped extra tight around those proteins. And when you're talking about your own personal genome, you'll talk about genes as being on specific chromosomes. That's because the chromosomes don't have any different information. It's the same DNA strand. It's just more compacted. When we're referring to a chromosome, we're referring to everything sort of like tightly packaged together. So here in our image, we can see what chromatin would look like. So you can see how it's loose and kind of windy and kind of messy. And here you can see when it's tightly packed into a discrete chromosome. Okay, so usually when you're talking about genetics though, you're thinking about genes. So genes are a specific part of DNA, so a specific piece of it that's going to code for a trait. And the way it codes for a trait is by coding for a specific protein. So protein can be something like a pigment that gives you hair color, or it can be something like an enzyme that controls a specific process within a cell. But a little piece of DNA is going to code for that specific protein that results in some sort of visible, measurable trait. And that's what our gene is. The gene is that stretch of DNA. So here on this image, this is a chromosome. So this linear sort of chromosome, stick-like, worm-like thing is our chromosome. And here you can see a gene highlighted. So here I'm pointing out in yellow, we can see one gene. So that would code for one specific protein. The reason you see two sort of wormy stick things in this image is that in diploid organisms, and humans are a diploid organism, every cell, every normal body cell has two versions of every chromosome. One that comes from each parent. So like one comes from your mom, one comes from your dad. And they're called homologous chromosomes. So these two paired chromosomes, they have the same sort of stretches of DNA on them. So they have these same genes that code for proteins. But when we're thinking about a gene, sometimes there are slightly different versions of code that can give you the same protein, or like similar enough protein that it works about the same in a cell. And these different versions are called alleles. So on two homologous chromosomes, so they're the same chromosome, you have numbered chromosomes like 1, 2, 3, 4. You have 2 of chromosome 1, 1 from each parent. You have 2 of chromosome 2, 1 from each parent. But they're going to be slightly different because they're going to have slightly different versions of each gene, which codes for slightly different versions of a protein. So these different versions are called alleles. So here we can see this yellow gene has a turquoise gene over here. So these are the same gene. So they code for a protein that does the same thing. So like 2 different pigment proteins, for example. So 2 different color proteins, say for brown hair or blonde hair or something. So they do the same job, but they do it slightly differently because the genes are slightly different. So the proteins are slightly different. So those are our different alleles on our homologous chromosomes. So here we see a picture of human chromosomes. You have 23 pairs. So numbers 1 through 22 are normal autosomal chromosomes. So we refer to those, basically they're chromosomes that aren't sex chromosomes. Your X chromosome, and if you're biologically male, your Y chromosome are sex chromosomes, so they're a little different. But you can see chromosomes 1 through 22 are the same length. They have similar sections, so these ones are dyed with different stretches, colored different colors. And that's because humans are diploid organisms. So we can see that you have 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3. They might contain slightly different code, but they do generally the same job in these diploid organisms. So those are homologous chromosomes that we're looking at here. Now, the caveat is that there are some specialized cells, which we call gametes. So they're basically sex cells. These specialized cells are haploid. So in a haploid cell, you have unpaired chromosomes. So you'd have like just 1 of this chromosome 1, just 1 of this chromosome 2, and so on. And they therefore only contain half the information, basically, because if you have, right, a gene for brown hair on chromosome 1 here, and a gene for blonde hair on chromosome 1, the homologous pair over here, if you suddenly only have one of those chromosome 1, you now have half the information, you only have one of those two alleles. So that's what a haploid cell is. But those haploid cells really only occur as we approach sexual reproduction. So those are going to be your sperm cells and your egg cells, but all of your normal body cells are diploid and have pairs of chromosomes, so homologous pairs. As we get into cell division, these chromosomes are going to look a little bit different, because in order to make two cells from one cell, we need to make a copy of our DNA. So every chromosome is going to get copied. It's going to get replicated to give us two identical DNA molecules that are just like that first one. So we started with one double helix, and then we're going to make two double helices that are copies of the first. When we copy a full chromosome, it produces what we call a chromatid. So we call these things chromatids, and they're basically like paired chromosome copies. They're going to be identical to each other if we're talking about one DNA molecule, one chromosome that was copied, and we now have two. Each of them is a chromatid, and eventually they're going to become their own chromosomes. But for a while in cell division, they're going to be kind of stuck together, creating this kind of X shape, which we're still going to refer to as a chromosome, because there's no additional information. It's just we kind of have double copies of the exact same alleles, the exact same genes. I'm sorry, that's kind of confusing, but that's why you'll see individual things that just look like a stick referred to as a chromosome, and then you'll also see this X shape referred to as a chromosome. That's because this is just one copy of our DNA here in our unreplicated chromosome. And this X-shaped chromosome, still a chromosome, it still just includes like one set of DNA, but it's copied over. So it's able to then split apart and send that information into one cell and into another cell. So we start with a chromosome, then we have two chromatids, which together in their X shape are still referred to as a chromosome. And then when those chromatids split apart, we're going to refer to them as chromosomes on their own in their new cells.